Hello, good evening. I am Surasree. I am a PMRF scholar working in the Department of Biotechnology, IIT Kharagpur, and I welcome you all to the week 4 live session of the NPTEL course Experimental Biochemistry. So in the previous weeks, we had discussed about the structure of amino acids, the titration of amino acids, isoelectric point. Then we also discussed about the structure of proteins and what is the peptide linkage. And we will, in this live session, in this week, we will look more into more details of protein structure and how the amino acids and the characteristics of the amino acids determine the structure of the protein, and what is the primary, secondary, tertiary structure of proteins. And also we will look at protein folding and protein denaturation. So how does a protein fold? And denaturation is when a protein unfolds. So we will discuss these topics today. All right, let's begin. So we had discussed about the amino acid and what is the general structure of amino acid. That is, it has a alpha carbon, which is a chiral carbon in most of the amino acids. Then it has one R group. So here suppose it is an R group, which is a side chain, and it has an amino group and it has a carboxyl group, right? So when two amino acids, they are condensed or they are joined together by the elimination of a water molecule, then they form the peptide linkage or peptide bond. So this is amino acid A, for example, and this is amino acid B. Okay, and here what you can see, that the OH of the carboxyl group and an hydrogen atom from the amino group of two consecutive amino acids, they join together and they leave as a water molecule, and the rest, so the C double bond O and NH, they are joined to form the peptide bond. So CONH, this is the peptide bond which is formed between two amino acids. Now when these two amino acids again join with a third amino acid, another peptide bond forms, right? So this is a dipeptide. What will be a polypeptide? Or first let's talk about what will be a tripeptide. What will be a tripeptide? A tripeptide will be formed by joining of three amino acids. Joined by peptide bond. And also there will be a polypeptide where many amino acids are joined together by peptide linkage, and a polypeptide is actually a linear sequence of multiple amino acids. Okay, now let's talk about the characteristics of a peptide bond. So CONH, I already told you that this CONH is the peptide bond. But how does this peptide bond behave? So this peptide bond is actually, you can see that it is a single bond between C and N, but it does not behave like a single bond. So it has both the characteristics of a single bond and a double bond. So it has both single bond and double bond character. So what do I mean by that? It is that it has a delocalized electronic system. So what is that? So you can see here that this nitrogen has a lone pair here. This particular nitrogen has a lone pair which it donates, and when it donates this lone pair, the oxygen can withdraw this lone pair towards itself, right? So because of this, it forms a double bond. It takes a double bond appearance, right? So this single bond therefore can also behave as a double bond, and because of this, what happens, because of this lone pairs, there is a delocalized electron system that forms. So this is the structure I'm talking about. So you can see that there is a delocalized electron system between the oxygen and the nitrogen, right? So this is why the peptide linkage does not have the characteristics of only a single bond or a double bond. It has both the characteristics of single bond and double bond, and also it is a planar structure. And if it was a single bond, or if it was a double bond solely, then its rotation would be different. But since it has both the characteristics of double bond and single bond, its rotation is restricted, and we will see what is its rotation later on. Okay. Alright, so we are talking about characteristics of peptide bond, and here we already talked about that peptide bond is formed by the condensation reaction between the carboxyl group and the amino group, and that peptide bond has a delocalized electron system. Now let's talk about the cis and trans configuration of the peptide bond, and which one is stable or which one is most observed. So trans configuration is when the two R groups, when two amino acids are joined together by peptide bond, these two amino acids have two R groups. So if these two R groups are located on the same side of the bond, then it is a cis configuration, and if these two R groups are located on the opposite sides, then it is a trans configuration, right? And of course a trans configuration is a more stable configuration. And why is that so? Because a trans configuration, in a trans configuration there is less chance of steric hindrance, whereas in a cis configuration the steric hindrance is much more. You can see here in a cis configuration the steric hindrance is much more. So which is why the cis configuration is less stable than the trans configuration. And obviously this stability and the steric hindrance will depend on the R groups. So it will vary according to the amino acids. So most steric hindrance, so proline has most steric hindrance, and the least steric hindrance is observed in glycine, because glycine, you know, it has an R group which is an hydrogen atom only. So of course it has least steric hindrance. Now, so this trans configuration of the peptide linkage is much more stable, and it is more common in peptide bonds. Now we will also talk about the torsion angles of the peptide bond. So these are the angles by which these bonds can rotate. Okay, so The first angle that we will talk about is angle phi. So this is phi, psi, and this is omega. These are the three torsion angles of a peptide bond: phi, psi, and omega. And phi is basically formed between N and C alpha. So the N of the amino group and the alpha carbon. So that is the angle which is phi angle. So the N and C alpha, this bond can rotate by this angle which is called phi. So the rotation of this bond can be measured by this phi angle. Now there is another angle which is formed between C alpha and the carbon of the carboxyl group. So the C alpha and the carbon of the carboxyl group, that is the angle which is called psi. So the C alpha to C, this C alpha to C, this bond can rotate by which angle? That is called psi. So and the other angle, which is the actually the rotation of the peptide linkage itself, the CONH, that is measured by omega. So we have three angles here. One is phi, psi, and omega. Okay. Now there are also these R groups which are attached with these C alpha, and you can see that here these two R groups are on the two opposite sides, which means that it is in a trans configuration, right? Now also it has to be remembered that this degree of rotation of these bonds are also depends on the R group, because the R groups are of various different types in different amino acids. So it can be a polar R group, it can be a non-polar group, and if it is polar, then it will always want to interact with the water, that is the medium, the surrounding medium of the protein, right? So if it is a polar group, then it will try to face outside or on the surface of the protein. So it will rotate accordingly. And if it is a non-polar group, then the degree of rotation or the rotation of R group will be towards the interior of the protein molecule, because it will not try to face the hydrophilic environment or the hydrophilic medium. So it will try to remain in the hydrophobic medium or the hydrophobic interior of the protein. So this degree of rotation of the R group also determines the 3D structure of the protein. So this is very important, because the 3D structure of the protein has to be such that the protein can function. So the structure of the protein is very essential for the function of the protein as well. So therefore this uh, 3D structure is determined by many other factors, as we will see later in this lecture, but also because of this presence of different R groups, the characteristics of these R groups, their ionizable properties, and also how they interact with the environment. So these all things determine the 3D structure of the protein. So now let's talk about the different terminologies which are associated with protein structure, which is a primary structure, a secondary structure, tertiary structure, and quaternary structure. Primary, secondary, tertiary, and quaternary. So what is the difference between these terminologies? So the primary structure of protein is nothing but a string of amino acids which are joined together by peptide bond. Okay, so this polypeptide chain is what is known as the primary structure of protein. So therefore it is nothing but a string of amino acids which are linked together by the peptide bonds. That is the primary structure. Now the secondary structure of protein, now when this string of amino acids, it takes up local folding into either a helix or a beta sheet or turns. So the secondary structure can be of different types. The most common ones are alpha helix, beta sheet, and turns. So this is local folding of this polypeptide chain. Now how is this folding determined? We will see later on, but remember that this folding entirely depends on the sequence of amino acids. Okay, so how a protein will fold, what will be its final 3D structure is determined by its sequence. And we will also see that this alpha helix sequence, beta sheets, they are also formed by several bonds which are formed between the amino acid residues. Now next, coming to the tertiary structure of protein. So what happens is that when these secondary structures, that is the helices, sheets and turns, when they again are folded together and it forms a gives a three-dimensional shape due to the interactions of these side chains or interactions of the R groups, that becomes the tertiary structure of the protein. So this is how a tertiary structure will look like, composed of the secondary structure elements but packed together in a 3D conformation. So this is called the native state of the protein also. And lastly, there is a quaternary structure also, where many polypeptide chains are joined together or are folded together. So this is a structure where many polypeptide chains or subunits, these are also called subunits of a protein, are folded together. So that gives the secondary structure of that gives the quaternary structure of the protein. And not all proteins will have multiple polypeptide chains, but the proteins those have which have those multiple polypeptide chains or the subunits, they are folded together in this quaternary form. So now let's talk about how this protein structure is determined. This protein structure is determined by various factors, which are rigidity of the backbone. So the backbone structure determines how the protein will fold. Interaction of the amino acids with water. So I told you that protein always remains in aqueous environment, and in aqueous environment how the amino acids are interacting with the water molecules, and also interaction among the amino acids themselves. So these interactions can be hydrogen bonds, these interactions can be electrostatic interaction, it can be hydrophobic interactions. So there are or it can be like bonds like disulfide linkages. So there can be multiple types of interactions between amino acids. So if like suppose both two amino acids, if both are charged and one is positively charged, another is negatively charged, then they will interact electrostatically. So these kind of interactions also determine how a protein will fold, and this information lies in the sequence or the amino acid sequence or the primary sequence of the protein or the primary structure of the protein, right? So now let's discuss in detail what are these several structures of the protein. Primary structure, I have already told you that it is a linear polypeptide chain, and there can be two types of linkages that can be seen in a primary structure. One is a peptide bond, which is obviously the principal interaction or the joining between two amino acids, right? All these amino acids are joined by peptide bonds, and there can also be presence of disulfide linkages. So between two cysteine residues. So cysteine, we already had talked about that the structure of cysteine involves sulfhydryl groups, SH groups, right? And when these two SH groups, they are joined together, it forms a disulfide linkage. So these linkages are observed in primary protein structure, and always remember that the primary structure of protein begins with an N terminal and ends with the C terminal. So what is N terminal, what is C terminal? So N terminal is nothing but the amino terminal. It is called the N terminal, and this is a standard that we maintain, that we begin structure of the protein or the sequence of the protein with an N terminal and end with the C terminal, which is the carboxy terminal. So begins with amino terminal, ends with carboxy terminal. So that is the primary structure of protein. Now coming to secondary structure, I told you that this is how the secondary structures look like. This is an alpha helix, and this is a beta sheet, or you can call it a beta pleated sheet. So now if you look at these detailed structures, the alpha helix looks like this. This is an alpha helix. So it's a helix form, right? And usually alpha helices are right-handed helix. So how is this helix formed, or how is this beta sheets formed? So the dominant interaction here is the hydrogen bond, formation of hydrogen bond. So you can see that if the hydrogen bond forms between the NH and the O of the carboxyl group, then there has to be a regular pattern in which these hydrogen bonds will form, then it can form a helix. Similarly, there has to be a regular pattern for a beta pleated sheet. We will look into the details of these hydrogen bonds. So this is how the secondary structure looks like, either a helix or a sheet, and if it does not have a determined secondary structure, then it can be a loop. Okay. Now let's see. Let's look at the alpha helix. Alpha helix, I told you that it is typically right-handed. Left-handed helices are rare. Now alpha helix is formed by the formation of hydrogen bond between the CO, that is the O of the carboxyl group, of residue I, and the NH residue of NH of residue I plus 4. So basically, if suppose there are 1, 2, 3, 4, 5, 6, 7. Suppose there are 7 amino acids. Okay. Now if there is a hydrogen bond formation between the carboxyl group of the first amino acid with the hydrogen from the amino group of the fourth amino acid. Right? And if this pattern is followed, then what happens is that this forms a helix kind of structure. So residue 1 with residue 1 plus 4, sorry, this should be 1 plus 4, which means this should be with the fifth residue. So, and similarly like this, there should be repeated pattern of hydrogen bond formation between residue I and residue I plus 4. Now, not only I and I plus 4, but also there can be different types of helixes where hydrogen bond is formed between I and I plus 3 and I plus 5. So there should not be any C. Okay, so that is called a 310 helix and a pi helix. So a 310 helix is where hydrogen bond is formed between residue I and I plus 3. So if there is a hydrogen bond between it should be between residue 1 and 4. In case of 310 helix I am talking about, between 2 and 6, sorry, not 6, 3 and 6, like that. Okay, so this is for a 310 helix. For a pi helix, the hydrogen bond should be formed between I and I plus 5. So 1 and 6, 2 and 7, 3 and 8, like that. So for a pi helix. And for a alpha helix, for a typical alpha helix, it should be between 1 and 5, 2 and 6, 3 and 7, right, like that. That is for an alpha helix. Okay, so the differences in hydrogen bond formation that determines the shape of the helix, and we are naming the helices by different terms. Now, in a typical alpha helix, there should be 3.6 residues per turn and 1.5 angstrom rise per residue of this helix. And there should be also 100 degree per residue turn. So this turn happens after 100 degree. The pitch is 0.54 nanometer, and there are several amino acids which either form the helix or break the helix. So what are these amino acids? So helix formers are alanine, glutamate, leucine, methionine. These are the amino acids which form the helix, whereas proline, glycine, tyrosine, serine, these are the amino acids which can break the helix. So if there is a proline, there will not be the alpha helix. So suppose there are 10 amino acids which are forming the alpha helix, and then there is a proline. So then at this point, if there is a proline, the helix will break. The helix will not continue any further, right? So the helix will not be formed any further. So that is why they are called helix breakers, whereas the amino acids which facilitate the formation of an helix are helix formers, which can be alanine, glutamate, leucine, methionine, like that. And there can be approximately starting from 4. So 4 is the minimum number of amino acids which should be present for formation of a helix, and there can be from like 40 plus residues also in a helix if it is a long helix. So that is an alpha helix. Now what is a beta sheet? A beta sheet is when hydrogen bond is formed between two strands. So here this is called a strand, and you can see the formation of the hydrogen bond between the CO and NH groups, right? And this particular structure is called a 4-stranded beta sheet. 4-stranded beta sheet, you can see there are 1, 2, 3 and 4 strands, and the beta sheets can also be anti-parallel or parallel. So what do we mean by anti-parallel or parallel? Anti-parallel is when the beta sheets are running in opposite direction. So for example, this and this strand, you can see that these beta strands are the strands are running in opposite direction, so they are anti-parallel. Again, with strand number 2 and strand number 3, you can see they are again anti-parallel because they are running in opposite directions. Whereas this strand number 3 and strand number 4, they run in similar directions, so they are parallel. So beta sheets can be anti-parallel or parallel depending on the direction, their direction. And beta sheets also have particular values of phi, psi and omega angles, the torsion angles, right? So phi, the value of phi is minus 120 degrees, psi is 120 degrees and omega is 180 degrees. So that is how the beta sheets are formed. Now how are these beta sheets and these alpha helices and the beta sheets, how they look like? So this is the structure of the helix. This is an alpha helix in a protein molecule, right? And these are the beta sheets. So they look like this kind of structures. So beta sheets, you can see that they form sheet kind of structures, whereas helices they look like helix, right? And there can be also turns. Right? So these are the prominent secondary structures which can be present in a protein. Now, scientist G. N. Ramachandran, in the year 1963, he figured out the geometrical features of a backbone and plotted the phi and psi angles in a plot which was later known as Ramachandran plot, which we commonly call as Ramachandran plot. So it can also be called a phi versus a psi plot. So what is done here is that in the y-axis you can see that the psi angles are plotted, and in the x-axis the phi angles are plotted. And there are several regions you can see in these Ramachandran plot, which are the possible regions of a beta sheet and alpha helixes. So possible regions means what? So basically these are the values which are possible. These phi and psi values are possible for these kind of secondary structures, either a beta sheet or an alpha helix. It can be a right-handed alpha helix, it can also be a left-handed alpha helix. So this plot shows that which conformations are possible for a protein, and by this plot we can also validate protein structure. How do we do that? Because we can see that the alpha helix, the 310 helix, pi helix, turn, parallel and antiparallel beta sheets, they have defined phi and psi values, right? The values of phi and psi angles, and these, when they are plotted in this phi versus psi plot, you can observe the regions. So suppose this is the region which is the probable region for beta sheets. So if your protein has beta sheet, then it will fall in this region of the Ramachandran plot. If it does not fall in these regions, if it is an outlier, then that conformation might not be possible for the protein, right? So that that geometrical features will not be present in a protein, right? So that is the significance of Ramachandran plots. We can validate the structure of a protein and we can determine which conformations of the protein are possible. Am I clear? Any doubts so far? Okay, if there is no doubt, then we might proceed. Alright. And here are these residues which are preferred for a helix, for a beta sheet, or for a term. Alright. Now let's talk about the next topic, which is the folding of a protein. So how do these proteins determine, or how do these proteins gain their 3D conformation or a native conformation? So the 3D structure is also called the native state of the protein, and the features of this native state is that it has a well-defined three-dimensional structure. It has a definite isoelectric point, and also it has a defined function. So this function is determined, or it is actually there is this relationship between the structure and function of the protein, which is called a structure function relationship. So the structure or the native state of the protein determines its function, right? So what is the driving force of protein folding? So it is hypothesized that the hydrophobic collapse is the driving force of protein folding. So what does it mean? So suppose as if you put oil in water, so oil and water separates, right? So one is hydrophobic, oil, and the other one is hydrophilic, which is water. So just like oil and water separates, similarly there are hydrophobic amino acids in the protein. So these hydrophobic amino acids will try to stay away from water, and what it will try to do is it will try to collapse, and it will try to be in close contact with other hydrophobic amino acids, which will together form a hydrophobic core of the protein, right? So therefore what will happen is that, or what is actually thought or assumed, that is that the hydrophobic amino acids, they fall together, and that is the driving force which actually drives protein folding. So that is hydrophobic collapse. Now after this, so this is an unfolded protein. So this is you can call a primary structure of the protein, where there is no particular folding which is observed. Then this primary structure of the protein, there are these amino acids which are coming close together, the hydrophobic amino acids. So that is called the hydrophobic collapse. And then what is happening is that there are this formation of secondary structures. So it can be helix, it can be beta sheets. So these secondary structures start to form, and then there is a state which is called a molten globule state, and this is an intermediate in protein folding. It is, you can see that it is not a native state, but it is very close to the native state. But remember, it is a more compact structure. It has all the secondary structure elements like the helix and the sheets, but still it is not the native state, and it is a compact kind of state. So this is called a molten globule state, and after which there is an appearance of the native state or the three-dimensional state of the protein. Now this protein folding process is a fast process, but there are other protein molecules like which are called chaperones. So these chaperones, they often enhance the speed of protein folding for some of the proteins. Okay, so these are called chaperones, but they do not alter the structure of the protein. They only speed up the process of folding. Now let's look into the thermodynamics of protein folding. So suppose if you have an unfolded protein, and then if the protein starts to fold, what will be the change in the Gibbs free energy? So we know this equation, that delta G is equal to delta H minus T delta S, right? So for a folded protein, or if we actually what we should do is that if we take the reverse reaction, that is when the protein is folding and not when the protein is denaturing. So when the protein is folding, so what is happening? The protein from an unfolded state is going to a folded state. Now this protein folding, is it thermodynamically feasible? So let's look at that. So an unfolded protein, in an unfolded protein, the delta G is a small negative value, and delta H, the delta H is a small negative value, and delta S has a large positive value, because in an unfolded protein there can be random number of conformations that can exist, so which increases the entropy. So the delta S value is large positive and delta H is small negative. Now when the protein is folding, then what happens? Then the delta H is large negative and the delta S becomes small positive. So in this way, so what happens here is that the delta S changes from large positive to small positive. So therefore we can say that the change in delta S, del S, is actually positive. So the del S is positive. So if del S is positive, then what we can say is that minus T del S, that will be negative. So this is a negative, this has a negative value, minus T del S. Now H, the enthalpy, it changes from small negative to large negative. So therefore the change in enthalpy or delta H will also be negative. So if your minus T del S is negative and del H is also negative, then obviously del G will also be negative. And we can say that since del G is negative, then therefore the process is thermodynamically feasible, right? So let's look at this graph, this particular graph, where you can say, where you can see that this is a graph of the reaction coordinate with the Gibbs energy, and here you can see that the unfolded state has a higher Gibbs energy than the native state, and to move from the unfolded to the native state, there is a energy barrier that is needed to overcome. So when the protein can actually overcome this energy barrier, this is called the transition state. So this is the delta G of folding, This is called the delta G of folding, right? So when the protein is folding, and this particular this value, this is called the delta G of unfolding. So when the protein from the transition state comes back to the native state, and this is the net delta G, which you can see is negative. So therefore the protein folding is thermodynamically feasible. And this is another equation by which you can calculate the value of delta G, which is the delta G of of the native state with respect to the denatured state, right? So this is nothing but minus RT ln K, and K here is written as the difference in concentration of the, or the ratio of concentration of the denatured state divided by the native state, or the ratio of concentration of the denatured state to the native state. So this is delta G equals to minus RT ln K, or here it is written as minus RT ln D by N. So this is the thermodynamics of protein folding. So of course when the reverse happens, that is when denaturation happens, then what happens, that the entropy becomes higher, and there has to be several interactions inside the protein which is needed to overcome, or which are required to be destabilized to unfold the protein. So what are these interactions? These are hydrophobic interactions, ionic interactions, hydrogen bond and disulfide bond. So these are the interactions that stabilize the folded state of the proteins. So these are interactions are between the amino acid residues, right? Hydrophobic interaction, ionic interaction, hydrogen bond and disulfide bond. So these interactions stabilize the folded state. So if this unfolded state has to be achieved, so that is, if the protein has to be denatured, then there has to be like these interactions have to be destabilized, right? So this is again showing you the change of del S and del H. So T del S from decrease in conformational entropy. So when the protein is folding, there is a decrease in conformational entropy. So that is negative of T del S, which is a very high value. Now minus T del S from, this is from the hydrophobic effect, which is favorable for protein folding. So this is the net T del S for folding, right, which has a negative value, and this is the del H for folding, which also has a negative value. So therefore the delta G for folding also has a negative value. So it is a spontaneous process. Now proteins are called marginally stable because the difference between the native and the unfolded state, the difference of delta G between native and unfolded state is only 5 to 15 kilocalorie per mole. So you can see that this difference is very low. So that is why they are called marginally stable. Now there are forces which favor protein folding. I already told you that these interactions, the hydrophobic interactions, hydrogen bond, ionic interactions, these are the interactions which favor the folding of the protein, whereas the principal force that opposes the protein folding is nothing but conformational entropy, because when the protein is unfolded, it can exist in various conformations which increases the entropy of the system. So any doubts so far? Okay, Alright, so now let's talk about denaturation of the protein. And what do I mean by protein denaturation? So protein denaturation is nothing but unfolding of the protein molecule. And this denaturation process is not necessarily complete unfolding of the proteins. And actually the protein can exist in multiple random conformations, which is not its native state. But the denatured state is also not the state where the protein will be functional. So the protein cannot function in these conformations, right? So that is the denaturation process. And denaturation is also a cooperative process. So which means that when there is denaturation, when it begins in one part of the protein, it accelerates the denaturation in other parts. So it is a cooperative process. And there is a particular method by which we can determine this cooperativity. We will see later on. So this denaturation process can be brought about by several agents. One can be heat, one can be extreme pH, denaturing agents like urea, guanidine hydrochloride. There can be several types of denaturing agents. The principal, very common one is heat. The effect of heat is that it destabilizes hydrogen bonding. And if it destabilizes hydrogen bonding, then of course the secondary structures, for example, which are formed by hydrogen bonding, the alpha helix, beta sheets, they will not be stabilized if we keep on increasing the temperature. So therefore, at high temperature, the protein will be unfolded or denatured. And this can be measured by this particular technique called differential scanning calorimetry or DSC. Because why? Because protein folding is an endothermic process. So by the heat change, by measuring the heat change, we can determine, or we can observe the thermal denaturation of the protein. Now all proteins are not denatured by heat. So for example, there are organisms which exist in various hot springs. So for example, a bacterium called Thermodynamics erythema, so, or Thermus aquaticus. These are organisms which can exist in very high temperature conditions. In these conditions, of course, since these organisms have to survive, their enzymes have to be functional in this condition, and enzymes are proteins, right? And we are talking about that this in case of if the protein is faces high temperature, then it is unfolded or denatured. So of course the enzymes of this kind of organisms which live in hot spring areas, their enzymes or their proteins cannot be denatured by high temperatures, right? So one example of that is polymerase, which we have isolated from Thermus aquaticus, which is called a Taq polymerase. So it is a DNA polymerase. So what it does is that it helps in DNA replication. So this particular protein, for example, this Taq polymerase, it is resistant to thermal denaturation. Now the next denaturing agent is extreme pH. So if the pH is changed, so if it is too acidic or if it is too basic, then what will happen is that if the pH is too low, then there will be protonation of the ionizing groups, right? And if the pH is very high, then there will be deprotonation of the ionizing groups. of the side chains of the amino acids. Now, if this kind of protonation or deprotonation happens, then of course there will be the charge, the amino acids will be either positively charged, or the amino acids will be also negatively charged. So if suppose the pH is very low and all the amino acids they exist in protonated forms, then this there will be a repulsion of these positively charged amino acids, and this intramolecular charge repulsion will be higher than the attractive forces like the hydrogen bonds and other attractive forces. So of course if that happens, then the protein will be unfolded. The protein cannot maintain its structure in this kind of condition, right? So for example, if a protein which is a basic protein, let's say a basic protein, that means its pi is greater than 7, right? So in this case, the many of the amino acids of this protein will be basic amino acids, so they will be positively charged. And if there is low, if we reduce the pH, then what happens? Then the all the amino acids will begin to become protonated, and if this continues, then what happens is that there will be a repulsion between these positively charged amino acids, and which will lead to unfolding of the protein. Now next, coming to the denaturing agents, which are urea or guanidinium hydrochloride. So what they do is that they usually cause preferential solvation of the unfolded state. So the unfolded protein state is more solubilized in this kind of solvents, urea or guanidine hydrochloride. So they denature the protein, and it destabilizes the folded state of the protein. There can be organic solvents, for example ethanol, which interferes with the dielectric constant of the protein. So that will also disrupt the native state. Now there can be several techniques which can be used for measuring the stability of the protein, or the unfolding of the protein. Observe the unfolding of the protein. It can be absorbance based methods, so absorbance spectroscopy, fluorescence spectroscopy. These are the things that we discussed in the previous session in the last week. So if you need to revise these absorbance spectroscopy or fluorescence spectroscopy, then you can go back to the week 3 video, which will be available in the course website as well as in my YouTube channel. Now circular dichroism, this is also another technique which is used for measuring this protein unfolding, and we can also measure the melting temperature of the protein. We will see later on. NMR, differential scanning calorimetry, urea gradient gels. So these are gels where there will be a difference in the migrating rates between the folded and the unfolded states. Catalytic activity. So it means that the protein, if it is an enzyme, it will not behave similarly if it is in folded form and it is in unfolded form. So in unfolded form or denatured form, it will lose its catalytic activity, by which we can also determine the protein folding, unfolding, viscosity of the protein, and the chromophores or the fluorophore probes. So if there is unfolding, the chromophores will be exposed on the outer surface of the protein, and the fluorophores also will be exposed on the outer surface of the protein if there is unfolding. Because if the protein is folded, the chromophores or fluorophores may be inside the protein structure, but which does not happen if the protein is denatured or unfolded. Okay. Next, we will talk about this protein denaturation curve, and this will be the last topic of discussion for today. So we have already talked about this equation, delta G equals to minus RT ln K, and here what we see is that with respect to the denaturing agent, with respect to the concentration of the denaturing agent, or with respect to the increase in temperature, how the absorbance or the fluorescence intensity or viscosity or the ellipticity of the protein is changing. So we plot it in the y-axis, absorbance, fluorescence intensity, the viscosity or the ellipticity, and the temperature or the urea concentration, that we plot in the x-axis, right? And we observe that when we increase the temperature, so there is an increase in the absorbance value. Why does it happen? Because we know this absorbance of the protein is due to the amino acids, the tryptophan, tyrosine and phenylalanine. So if these amino acids are exposed on the outer side of the protein, then their absorbance will increase. And why will they be exposed on the outer side of the protein? Because the protein is unfolding, because we are increasing the temperature. So as we increase the temperature, more and more tryptophan, tyrosine and phenylalanine will be exposed to the outer surface, which will increase the absorbance of the protein. So that is why we can see this kind of a graph with respect to temperature, increase in temperature and increase in concentration of urea. Also we can see a similar kind of a graph, right? So that is our denaturation curve, or the unfolding curve of the protein. Now if we plot fractional denaturation, so that is how much denaturation is happening with respect to the temperature or urea concentration, then the highest value will be 1 when the protein is completely denatured, that is this particular state, and when the protein is not denatured at all, that is the value 0. So at the middle of this graph, therefore, that is at the value of 0.5, where the fraction of protein which is denatured and the fraction of protein which is in its native state will be equal, right? So if n equals to ft, so the fraction of protein in native state is equal to the fraction of protein in denatured state, and that 0.5 fractional denaturation, or this particular condition Fn equals to Fd, is achieved at this temperature, which is called the melting temperature. So this is the temperature at which this 0.5 fractional denaturation is observed. That is called the melting temperature of the protein, right? So at which 50% of the protein is denatured and 50% is in its native state. So when we are talking about this denaturation protein analysis of this thermodynamic analysis of denaturation, we always have to remember that this denaturation and folding, these processes have to be reversible, right? So this has to be a reversible process, and the equilibrium has to be reached very quickly for the thermodynamic analysis. Now we can also, as I told you, that this denaturation is a cooperative process, and we can have an idea of this cooperativity. How do I have an idea of this cooperativity? So if we plot delta G with respect to the concentration of urea or with respect to the temperature, for example, then what happens, that if we decrease the concentration of urea, so suppose we usually use 8 molar of urea, right, for denaturing the proteins. So from 8 molar of urea, if we decrease the concentration to 0 molar of urea, And if we determine the change in delta G value, and if we plot it in this kind of a plot, then at 0 molar urea, it is basically water, right? So there is no urea, it is only water. So this will give you the delta G of denaturation in water, right? And this will be the equation. So delta G, from this graph we can determine this equation. Delta G of denaturation is equal to delta G of denaturation in water minus m into concentration of the urea. So here this m is nothing but the slope, right? Slope of this graph, and this m will give us an idea of the cooperativity. And what is that? So basically, how much percentage of protein is denatured at what concentration of the urea? How much percentage of protein is in its native form? That is determined by this cooperativity. Okay, so that also we can determine from this denaturation analysis by these techniques like absorbance, fluorescence, CD, or differential scanning calorimetry, etc. So any doubts so far? Okay, the third graph is when you plot the change in delta G with the concentration of urea. So is in your x-axis it is the concentration of urea, and urea is in decreasing concentration. So so actually what you do is you take the protein in 8 molar urea, for example, which is standard used for protein denaturation. So you take the protein in 8 molar urea, and then you gradually decrease the concentration of urea. So from 8 molar you finally go to 0 molar of urea. So what is 0 molar of urea? If there is no urea, and what does it mean? That it is only water, right? So by this gradual decrease in the concentration of urea, we can determine the delta G value, which we will determine from this equation, right? That delta G is equal to minus RT ln yn minus y divided by y minus yd. So what is yn? It is the anything that you are measuring in the y-axis. It can be absorbance, it can be fluorescence intensity with respect to the native state. And what is yd? It is this same parameter, either absorbance or fluorescence intensity, with respect to the denatured state, right? And y is the value of this absorbance or fluorescence intensity at any point of time, right? So suppose first what you do is you decrease the concentration of urea from 8 to 7 molar, right? So when you decrease the concentration of 7 molar, then what you do is you calculate this value of delta G from the absorbance, for example. So what you do is you take the absorbance at this particular point of time, right? And you already know what is the absorbance of the native protein and what is the absorbance of the totally denatured protein, right? So you put these values in this equation and you calculate the value of delta G, right? So at 7 molar urea concentration, then what you do is you again decrease the concentration of urea to 6 molar. Then what happens? This then again you calculate the value of delta G from this equation again. Delta G equals to minus RT ln yn minus y divided by y minus yd, right? Now gradually you again keep on decreasing the concentration of urea from 5 molar to 4 molar to 3 molar to finally 0 molar. So 0 molar means the protein is in water, right? And how the protein behaves in water. So basically, what is the delta G value of the protein is in water? That is this value when there is 0 molar urea concentration. Now what you do is that you plot this delta G values with respect to the urea concentration in x-axis and the delta G values in the y-axis, right? And when you plot this graph, you can see that this kind of a linear graph forms, and therefore the equation you can put in the that delta G of denaturation will be equal to delta G of denaturation in water minus m into concentration of urea, right? Because it's a linear equation, right? Y is equal to, it takes this form of y equals to mx minus c, right? Or minus mx plus c, right? So therefore you can say that from this m, so m will be actually the slope of the graph, and this from this m you can have an idea of the cooperativity of the protein denaturation. Alright, then let's move on to the question answers. Please tell me your answers, or you can write in the chat box. Right answer. Next question. Correct. Correct again. Peptide bond is formed by condensation reaction. Next question. Are you sure? Yes, actually the answer is D, because first of all this is a tripeptide sequence. Okay, so tripeptide means what? There are 3 amino acids. So A, B, C suppose. So this is a tripeptide sequences, and what are the number of all possible tripeptide sequences? So for position A there can be 20 amino acids, for position B there can be 20 amino acids, and for position C also there can be 20 amino acids. So 20 into 20 into 20, or 20 to the power 3, that is 8000. That is the total number of possible tripeptide sequences that can be present. Okay, next question. This question has multiple answers. Okay, C is correct, but what if you have another answer, another option which is correct? No, not B, but C and D. So this double bond single bond character, this is due to the delocalized electron system. So C and D are the correct answers. Okay, next question. Okay, all right, correct answer. This question is about the torsion angles. Can you answer? Okay, the phi and psi angles I have explained previously. Suppose I think you hadn't joined back then. Okay. So these are torsion angles. So what are these torsion angles? So these are basically the angles by which the different the different linkages in the peptide bond, how can they rotate or how can they move. So the angles by which they can move is the other torsion angles. So the phi angle is between N and C alpha. Between N and C alpha, the angle which is present is called the phi angle. That means the bond between the N and C alpha, this particular bond, right? This can rotate by an angle which is phi. It is denoted as phi. The angle between N and C alpha, right? And the bond between C alpha and C double bond O, or which is the C alpha and the carbon of the carboxyl group, that is called the psi angle. This is denoted by psi. Okay, so the bond between C alpha and the carbon of the carboxyl group, it can rotate by an angle called psi. And the other one, which is the peptide linkage, so this one is the peptide linkage, right? The CN bond. So it is the bond between the carboxyl group and the next amino group. So this peptide bond or the CN bond, it can rotate by an angle called omega. Okay, so these are the torsion angles, phi, psi and omega. And these torsion angles, when they are plotted, psi, so this is how it looks like. This is called a Ramachandran plot, which is a psi versus phi angle plot. And there are defined values of the beta sheets and the helix, left-handed and right-handed helix, and there are defined values of these secondary structures of the phi and psi angles. So that means their rotation is restricted by these angles, right? The rotation of these phi and psi bonds. So again, going back to it once more, the NC alpha is the phi angle and C alpha to C is the psi angle and C to N is the omega angle. Is that clear? Okay. So the correct answer is that phi between NC alpha and psi between C alpha to C of the carboxyl group. Okay. Next question. Yes, D is the correct solution. Next question. So you have to think about which amino acids are helix former and which amino acids are helix breakers. So as we know that proline is a helix breaker. So if proline is present, then the helix will break. So this methionine, cysteine, glutamine, arginine, serine, they form an alpha helix, but the helix breaks when there is proline. So the possible sequence of the alpha helix will be A, methionine, cysteine, glutamine, arginine and serine. Correct answer. The next question. Right. Right answer. Next question. Are you sure? The correct answer is all of these. Because urea is a solvent which displaces the water molecules around the protein. Heat ruptures the hydrogen bonds and the hydrophobic interaction is also destabilized. So all of these is the correct answer. Next question. So this is a bit tricky question. Please think carefully. Okay. Can you explain why did you choose this option? Okay, so protein X, it is a basic protein as you already said, right? So because it's pi, you can see that it has a pi in the basic range, whereas protein Y is an acidic protein. So of course, protein X, when it is in pH 7, so the basic amino acid residues will be positively charged. So you can say that this is positively charged at pH 7, or the positively charged residues are present in this particular protein, right? And when the protein and in case of protein Y, the protein Y is an acidic protein, so there are like negatively charged residues, right? So therefore the negatively charged residues or the acidic residues are present in protein Y. Now you are adding glacial acetic acid, so it's an acid, so it will reduce the pH. Now we know when pH is reduced at low pH condition, there is protonation of the amino acids that occur. So the amino group is protonated, the carboxyl group is protonated, the R group, which will be positively charged in case of protein X, will also be protonated, right, in case of protein X, because at low pH there is protonation. Whereas in case of protein Y, the R group will be negatively charged, right? So therefore there will be less charge repulsion in protein Y when the pH is getting reduced, right? So in case of protein X, the unfolding will occur at a much higher rate, because there is more positive charge repulsion in protein X when the pH is lowered, right? At a point of time, the protein Y will also have all protonated residues, but that will occur at a less speed or at a lesser rate than protein X. So protein Y will unfold at a slower rate as compared to protein X. That is the correct answer. Okay. Okay, next question. Correct. C is a correct option, but also B will also be a correct option. So if it has multiple options, right? The unfolding of the protein will be causing this exposure of the tyrosine tryptophan residues. So B and C both are correct answer for the previous one. Now next question. Right, melting temperature. And the final question for the day. Right. So iodoacetic acid is not a denaturant of proteins. Alright. So with that we end the discussion of this week 4 of experimental biochemistry. If you have any doubts, please reach to me. You can send me emails, you can send questions in the discussion forum, and if you want the recorded lectures, you can visit this YouTube page, and also the links to all the YouTube recordings will also be present in the group, in the course website. So if you have any other doubts, please let me know. If not, then we will end today's discussion here, and thank you all for joining. We will meet again next week, this time Tuesday 6 to 8 p.m. Thank you.